Hello and welcome, my gentle and of course very modern apes. We've got some cool new information to talk about today. When you ask someone what the difference is between a monkey and an ape, usually what they'll say is that monkeys have tails and apes do not. Little kids can comprehend this distinction, and while it's ultimately wrong because technically apes are both anthropoid and catarrhine monkeys taxonomically, it is an important observation. Why is it that hominoid primates like humans, gibbons, chimpanzees, gorillas, and orangutans don't have tails? Some of you out there, these ones, are quickly pointing out that we actually do have postanal tails. They're just internal and they've been exapted for new functions. And at this moment, I would like to say to you, quit being a know-it-all. That's my job. But hominoids don't have tails, and as a youngster, and still to this day a little bit, I think that kind of sucks. I wish humans still had tails. That would be pretty awesome. I'm not thinking about the kind of tail that you might be thinking about in your head right now, like the furry kind that you put on a belt, like a neon wolf tail. That's not my vibe. I'm thinking more of like a grotesque prehensile tail. I want the kind of tail that some atelids today have, with a hideous leathery underside that allows them to have a nice grip while they're moving through the trees and foraging. Prehensile tails with calloused undersides have shown up multiple times in the animal kingdom over the course of mammalian evolution, just not hominins. And functionally, this makes just a load of sense. Look at these goobers try to stand bipedally. But tails, especially prehensile ones, would get in the way of our current locomotor style, which is a pretty efficient version of bipedality. Looking at the structure and form of the human pelvis as it is now, extending this coccyx much further would be problematic to say the least, not just for our locomotor style, but even for things like childbirth. So it is not at all surprising that a tail did not return in hominin evolution, so the organisms that exist moving forward in time towards Homo sapiens' spot, temporally speaking, after our split with chimpanzees. The reason that I've kind of sidestepped here and pointed out how tails would encumber a hominin when moving bipedally, and then gone on to point out that tails didn't reappear within hominins for this reason, is because tails were already long gone well before bipedalism shows up on the scene. And this is contrary to a line that appears in the paper that we're going to be looking at today, which proposes that one of the hypotheses for the loss of tails in hominoids is bipedalism, and I do not think that there is the scantest support for that. This paper is a masterclass in exploring the how of hominoid tail loss, but I really think it is imperative that we keep in mind the potential why for tail loss as well, because it is the selective pressure that caused this character to move to fixation in ancestral hominoids. What I'm saying here in this lengthy preamble, right, is that this paper is going to be explaining the how, and I am going to be explaining the why, at least to the best of our understanding at present. The paper that we're going to be looking at today by Gia and colleagues is exploring the genetic and developmental reason that hominoids do not have tails, that we do not grow them in utero. What has changed genetically in our blueprints, as it were, to prevent us from growing and developing tails, whereas our cercopithecoid colleagues do, for the most part, as we'll explore a little bit more in a moment. Ape evolution began in the Miocene, which is a time period that begins roughly 23 million years ago. And the primates that dominated the scene prior to that, at least according to the fossil record snapshot that we have from the Fayum in Egypt, was mostly a group of organisms that consist of parapithecoids, propliopithecoids, oligopithecoids, and proteopithecoids. And of these guys, we have one major player that is extremely well characterized, and this is a monkey called Egyptopithecus. Now, Egyptopithecus was pretty similar to modern cercopithecoids, right? It's a baboon, Gwynnon-looking thing that is pretty generic as far as what we think of when we think of monkeys, and most importantly, this thing has a tail. Now, the latest Egyptopithecus shows up approximately like 29-30 million years ago, whereas our first apes are showing up in the very earliest portions of the Miocene roughly 23 million years ago in the likes of proconsul Mesue. Now, you can put a big fat asterisk on proconsul Mesue being the oldest ape at approximately 23 million years of age, because there is a potential contender aiming to steal that spot in an ape, or I should say maybe ape, called Camelopardalis, whose dating range is 24 to 27 million years. Now, Camelopardalis is really poorly represented, so we aren't really 100% sure if it's an ape or not. It's mostly like dental samples, so we will see, and that's also pending more excavation of locations where Camelopardalis is found. It might be an ape, it might not, but what you need to know for now is that for sure, by 23 million years ago, we don't see tails in the hominoids. 23 million years ago, tails are gone. Bipedalism in hominins evolved 7 million years ago if you take Sahelanthropus chilensis as a potential habitual biped, and you can also put a huge asterisk on that. Our first for sure part-time biped would be Ardipithecus ramidus at 4.4 million years ago. So what I'm trying to say is tails disappear long before bipedalism evolves in hominins. So the idea that the loss of tails in ancestral hominoids was facilitated by a tendency towards bipedalism. Terrestrial bipedalism is typically what they mean when they say bipedalism. They're not talking about the type of bipedalism that you see in gibbons when they run across the branches without using their hands as anchor points, or the type of bipedalism that they propose for like Danuvius, a Miocene hominid. And then there's the other more obvious reason why that hypothesis doesn't really make a lot of sense, and that's that other hominoids who also lack tails are not bipeds, right? Chimpanzees aren't facultative bipeds, neither are gorillas, neither are orangutans. Gibbons are facultative bipeds when they come to the ground, but they do that so rarely that we mostly just consider them brachiators. And then, of course, on top of that, we have the first hominoids lacking tails not being bipeds either. So if it wasn't a pressure towards being more terrestrial, being more bipedal, or a combination of both, then what did cause the loss of tails in ancestral hominoids? Well, to figure that out, we should go ahead and take a closer look at the difference in postcrania between Egyptopithecus, a monkey with a tail, and some of the first apes, Merodopithecus, Proconsul, and Akembo. First, let's consider some pretty big differences in the functional locomotor complexes of modern monkeys versus modern apes. And to make this extremely simple, I'm going to describe it like this. Most monkeys today, whether they are arboreal or terrestrial, have skeletons that are more inclined towards stability and agility, whereas most apes have skeletons that appear to be more adapted towards mobility and strength. Compared to your average monkey, and when I say monkey, I am, of course, referring to non-hominoid catarrhines, most monkeys are not very flexible. A baboon cannot raise its hand above its head like you can. Neither can a macaque. They cannot dangle directly below a branch and maneuver deliberately through the trees using each limb as a point of contact. Monkeys are fast. They zoom through the trees for the most part above the branch, and they do so quadrupedally, on all fours. The fastest ape in the trees is a gibbon, and they do so using brachiation, which relies on their highly mobile shoulders. Extremely flexible limbs in the arms and the legs are characteristic of all the apes though, even if gibbons are the fastest. Orangutans, gorillas, chimpanzees, and humans, when we're in the trees, we are slower, more deliberate, but because we can use all four of our limbs as a point of contact and not just below our bodies, we can get to almost any part of the tree and we can do so above or below the branch. This is something that most monkeys can't do. Notable exception in the atelids, but we will get to them later. So the generalized ape skeleton is inclined towards greater mobility and potentially allows more feeding opportunities. But limb flexibility is not the only part of this complex that matters. On your screen now, you can see an ape engage in below branch suspension. This ape is clearly very athletic. Her mobile scapulae and wrists allow her to hang below the branch, while powerful grip strength allows her to hold her entire body weight using just one limb. Multiple muscle groups allowed this ape to pivot from one limb to the other while still hanging, or hoist her body upwards. What an impressive creature! Akembo hesloni, which is the oldest ape for which we have essentially the entire skeleton, has long been characterized as pretty monkey-like in its locomotor style. However, the word pretty there is doing some heavy lifting. Akembo hesloni's skeleton, especially the postcrania, shows some remarkably derived characters. Akembo's humerus, the bone of the upper arm, has a proximal portion, that is to say, the area closer to the trunk of the body, that for the most part looks more like monkeys, especially in the torsion or the degree in which it twists. The distal end, however, looks a lot more like apes, with an expanded trochlea and a rounded capitulum. At places like Rusinga, humeral heads have been found for genus Akembo. The humeral head is just the rounded proximal portion of the humerus, and this suggests that Akembo probably had extremely mobile shoulder joints. And what that means is that it was probably much more like apes than modern monkeys, and that it could probably hold its arms above its head. Like modern monkeys, Akembo had 6 lumbar vertebrae and an extremely narrow pelvis, which suggests the sort of deep, narrow chest and hips that you tend to see in modern monkeys, to the exclusion of modern apes, which have 4 to 5 lumbar vertebrae and more barrel-like chests. There are aspects of the lower limbs, however, that look much more like apes than monkeys in Akembo, and in Proconsul and Merodopithecus as well. This is specifically the angle of the femoral head, which suggests greater mobility at the leg than modern monkeys tend to have, and additionally, features of the patella suggest that this animal was using its legs for deliberate, powerful motions rather than the quick springiness that you tend to see in arboreal monkeys that are running on the tops of the branches. Many of the features found in Akembo are found in Merodopithecus as well. Merodopithecus actually had additional derived characteristics that align it even more with modern apes in its lower back, which was stiff and stable as compared to monkeys, and suggests, in addition to what we see in Merodopithecus's femur, that what it was doing was a lot of vertical climbing. Modern apes do not climb like modern monkeys, due in part to their much larger size, but this holds true even for gibbons. There's obviously a lot of biomechanical minutiae going on in the differences between the way that apes vertically climb and the way that monkeys vertically climb. But to simplify it, apes are slower but stronger, and monkeys are faster but weaker. It's no surprise that early morphologic changes that facilitate vertical climbing and greater grip strength, which are both precursors for the final ape condition, show up around the same time as tail loss. I think that grip strength probably came first. I think you had like a lineage of Aegyptopithecus-like things that had tails that, through natural variation, acquired stronger grips. Now, here's the thing about tails. The primary purpose of tails in these above branch arboreal quadrupeds, like baboons and macaques, is they allow the animal to balance itself. Particularly when they're jumping. They can right their position in the air. Increased grip strength allows an animal to feed just a little bit more terminally on the branches, and when in combination with slightly more mobile limbs, allow you to feed more terminally still. Feeding at the terminal ends of branches is not something that you do quickly or recklessly, which means you're already favoring mobility and slow movement over that quick springiness that you tended to see in the monkeys. In a few words, you're trading speed, agility, and stability for more food by being slow and mobile. Given what we know about the purpose of a tail, what do you need a tail for at this point? Now you might be thinking, oh, gutsickgibbon, grip strength and vertical climbing facilitating tail loss, that sounds a little bit just so. And to that I say, it would sound that way if it hadn't had happened twice just in primates. You see, in primates, there exists another interesting dyad of tailed above branch quadrupeds and tailless slow clamberers, and that is the comparison of two lorisoids. On the tailed above branch quadrupedal side of things, you have bush babies, and on the tailless slow clamberers, you have the lorises. The common ancestor of lorises and bush babies is thought to have looked a lot more like a bush baby than a loris, and what this would mean is that lorises have lost their tails as compared to their common ancestor with the sister group, much like apes have lost their tails as compared to monkeys, our sister group. as it turns out, the vertical climbing kinematics of lorises has been examined and analyzed in comparison to other primates, and they're most similar to Asian apes like orangutans and gibbons. And you know what else lorises are known for? Their grip strength. To put a little cherry on top of this idea, this hypothesis for the loss of tails in hominoids, I want to look at another kind of counterintuitive example, and that's the spider monkey that we talked about earlier. Genus Ateles, the spider monkeys, are interesting for a lot of different reasons with regard to what we're talking about today. For one, Ateles actually vertically climbs a lot like apes do. Their kinematics looks like the ape kinematics for vertical climbing, and this seems a little counterintuitive, right? Because they've got those giant tails. For two, they're also careful clamberers and sometimes brachiators like gibbons. In fact, their locomotor repertoire looks a lot like gibbons in general, except for one little problem. When gibbons feed, they hang by one hand below the branch and then they use their other hand to harvest fruits or leaves, whereas spider monkeys do something similar, except they've got one hand above and then they're also holding on with their prehensile tail. So both modern apes and modern spider monkeys are slow vertical climbers and clamberers that sometimes brachiate. That's their locomotor repertoire for foraging their food. Except spider monkeys have a tail, and it's not just a little tail, it's a big-ass tail. How then can we posit that it was a change in the locomotor repertoire of ancient apes that led to the loss of the tail? Well, baby, it's grip strength. Here's the hands of 5 modern apes. They're known for having powerful grips, due in part because of a powerful opposing thumb. But genus Ateles doesn't have a powerful ape-like grip. And do you know why? It's because genus Ateles doesn't have any thumbs. So what I'm saying here is that it's convergent. The slow vertical climbing and clambering that we see in atelids like spider monkeys and that which we see in hominoids are both achieved through different means. In hominoids and in lorises, this method of locomotion and foraging is facilitated by a powerful grip. This in and of itself made tails kind of pointless, and so the tails were lost. On the other hand, what we see in the atelids is an arrival at the same conclusion, but instead of losing the tail, they actually lost the grip and increased the mobility of the tail, which effectively acts like a 5th limb. Slow vertical climbing and clambering opens up the terminal branches of trees to foraging, but it requires mobility and multiple points of contact in order to do so safely. In the case of hominoids, they achieved a greater grip strength and tails became redundant. In the case of New World monkeys like atelids, they achieved a prehensile tail, a 5th point of contact, and grip strength became redundant. Both groups also evolved mobile limbs in the form of flexible wrists and shoulders, as well as an orthograde or upright posture since they're hanging below the branches for much of their feeding time. But the inciting incident, the inciting feature for each group to move to this new style of foraging and locomotion was different. That's so cool. I think this is a super strong hypothesis with regard to why hominoids lost their tails, not the least of which because it draws from multiple lineages. To be clear, this isn't the only reason why a lineage might lose a tail or gain greater grip strength or gain a prehensile tail, but I think it makes a lot of sense in the context of the lineages that we're looking at. With that in mind, how does something like this happen? Mechanically speaking, how does this go down? How do you go from a population of tailed critters to a population of tailless critters? Is it gradual like we see with some characteristics, like the migration of the blowhole or nostril in cetaceans? Or is it like one and done? Like over the course of one generation, you have an individual that's born and they're just tailless? The answer may surprise you. Our paper today is fortunately open access so we can look at as much as we desire. It's titled On the Genetic Basis of Tail Loss Evolution in Humans and Apes by Gia and colleagues, and like I said earlier, it was published this previous February, and in fact, it was published a couple days ago. That being said, there has been a preprint available for this thing for several years now. We've been waiting for bated breath for this bad boy to come out. So, as usual, we will start with the abstract. The loss of the tail is among the most notable anatomical changes to have occurred along the evolutionary lineage leading to humans and the anthropomorphous apes, with a proposed role in contributing to human bipedalism. The only way that the tail loss could contribute to human bipedalism is kind of potentially predisposing an organism like a tailless ape to be bipedal. So maybe that's what they were going for. It's kind of occurring to me now that maybe that's what they were going for the whole time. Even if that is the case, I think they should have specified, because I don't believe they do later in this paper. Yet, the genetic mechanism that facilitated tail loss evolution in hominoids remains unknown. Here we present evidence that an individual insertion of an ALU element in the genome of the hominoid ancestor may have contributed to tail loss evolution. So an ALU is just like a repetitive DNA element. That's probably a simplification, but that's really all you need to know for now. We demonstrate that this ALU element inserted into an intron of the TBXT gene pairs with a neighboring ancestral ALU element encoded in the reverse genomic orientation and leads to a hominoid-specific alternative splicing event. We're going to break this down in a minute. To study the effect of this splicing event, we generated multiple mouse models that express both full-length and exon-skipped isoforms. So an isoform is just a type, right? So they're basically saying we tried this ALU's impact in its like partial form and in its full form, if I'm understanding this correctly, mimicking the expression pattern of its hominoid ortholog. This is because ALUs are primate specific typically, so they went to the mouse model and they found like the mouse version of what's going on in hominoids, and they kind of like inserted it. They inserted a version of what we see in hominoids into mice to see if they're born tailless. Mice expressing both TBXT isoforms exhibit a complete absence of the tail, or a shortened tail depending on the relative abundance of the TBXT isoforms expressed at the embryonic tail bud. So I'm assuming this just means there was variation in like where the expression happened, and if it was like at the very terminal end of the developing tail bud, then no tail, and if it was maybe off to the side a little bit, you might get a little bit of tail formation. These results support the notion that the exon-skipped transcript is sufficient to induce a tail loss phenotype. Moreover, mice expressing the exon-skipped TBXT isoform develop neural tube defects, a condition that affects approximately 1 in 1,000 neonates in humans. Thus, tail loss evolution may have been associated with an adaptive cost of the potential for neural tube defects, which continue to affect human health today. TBXT is a gene that is associated with tail formation in primates. The authors of this study found that a specific ALU found in an intron of TBXT interacts with a neighboring ALU not found in the intron of TBXT, and that together they cease tail formation in hominoids. So all primates have the tail formation gene, but there's funny business going on in the hominoid genome with these specific ALUs interacting with one another that like stops the tail formation from actually happening. This was theoretical, like just looking at the genome, it seemed intuitive to say, given what we know about genetics and how all of these elements are interacting with one another, this is probably what's causing tails not to form in hominoids, in the apes. But these authors took it a step further, and they took the ortholog, so the gene that is similar, analogous to what's going on in primates, the TBXT gene, they found that in mice and then basically did what we see in the hominoids to some model mice. In the body of the paper, they say examining non-coding hominoid-specific variants among the genes related to tail development, we recognized an ALU element in the 6th intron of the hominoid TBXT gene. This element had the following notable combination of features. 1, a hominoid-specific phylogenetic distribution. So this version of the TBXT gene, while TBXT is found in all primates, this version was only found in hominoids, which is what made them go, maybe it's related to the fact that hominoids are tailless. 2, presence in a gene known for its involvement in tail formation. Again, they knew it had to do with tail formation. And 3, proximity and orientation relative to a neighboring ALU element. First, this particular hominoid-specific ALU element is from the ALU-Y subfamily, a relatively young but not human-specific subfamily shared among the genomes of hominoids and Old World monkeys. Moreover, the inferred insertion time, given the phylogenetic distribution, coincides with the evolutionary period when early hominoids lost their tails. So So guys, that is critical, and I feel like not enough people are harping on this. The molecular clock expectations of this gene match what we see in the hominoid fossil record that we just spent 15 minutes discussing. Yet again, evolutionary theory meeting the gold standard of science. Second, TBXT encodes a highly conserved transcription factor crucial for mesoderm and definitive endoderm formation during embryonic development. So it's at play during development in utero. Heterozygous mutations in the coding region of TBXT orthologs in tailed animals such as the mouse, the Manx cat, dogs, and zebrafish lead to an absence or reduced forms of the tail, and homozygous mutants are typically non-viable. So that's really cool too. Orthologs in other species, like cats for example, when they have something similar to the hominoid condition, they are also born without tails. There's a lot of really cool and frankly above my pay grade genetics stuff here, including a concrete description of like what's going on in the hominoids specifically. And then we get to the part that I think is really cool and like visually provocative, which is the picture of all of the mice without tails. So here in figure, which figure is this? In figure 3, we see the TBXT exon 6 isoform is sufficient, like on its own, to induce the tail loss phenotype. So A, B, C, and E here, D and E, are showing us the consequences of taking the hominoid condition and imposing it on tailed organisms such as mice. So D here, we've got a picture of the two little newborn mice, one with this tiny little stublet of a tail, really no tail there, right? There's no sacral or coccygeal vertebrae present at all, which is like actually a more extreme condition than what we see in hominoids, where we have sacral vertebrae, our sacrum, and coccygeal vertebrae, our coccyx. We just don't have anything further than the coccyx. So really, I think what we're looking at as the best analog for the hominoid condition is actually this one here that has 2 to 5 caudal vertebrae, because we have 3 to 5 caudal vertebrae, right? They're just all fused into a single bone, the coccyx. Now, the fact that there's so much variety here that encompasses both more extreme versions than what we see in hominoids and more conservative versions is explained here in figure 4, which notes that introducing inverted intronic sequence pairs induces short-tailed phenotypes in mouse models, phenotypes meaning more than one, and they do elaborate more on this in the body itself. Abundance of TBXT isoforms explains tail phenotypes. The next paragraph notes that there's like a lot going on here, and that it seems like we see a lot of different possibilities for other regions of the genome as far as these ALUs interacting with one another go. They talk about other signs here, saying the human genome contains 1.8 million copies of short interspersed nuclear element signs, including 1 million ALU elements, of which more than 60% are intronic. So there's like great possibility here for finding potentially more of these interactions. They want to systematically search for more of them to see the functional roles that they play in development and in disease. And they basically say that the results of the mouse work in particular demonstrate a correspondence between the relative abundance of different isoforms of that TBXT gene and the tail length phenotypes that we see not just in mice, but like other mammals. Why do we see so many different tail lengths? The gene responsible for tail length in mammals is sensitive to minute changes. They actually bring up lorises, which I think is interesting. They're like, yeah, we should look at the genes for lorises and see how analogous their situation is to hominoids, right? Because this was an independent tail loss in each lineage according to similar pressures for changes in locomotor styles. But it's very unlikely that they changed in exactly the same way. They also talk about cercopithecoids that have lost their tails, so mandrills and drills, Mandrillus sphinx and Mandrillus leucophaeus are both species that have greatly reduced tails, as well as Barbary macaques and Macaca sylvanus. And the reason I didn't bring them up earlier is because like they very clearly have lost their tails under a different type of selective pressure, probably one related to the degree of terrestriality that they're committed to, as well as potentially the climate that they live in. There's also Japanese macaques, so Macaca fuscata, where having a tail is not ideal when you're living in such freezing, or in the case of Barbary macaques, more temperate climates. We'll read the last paragraph to sum it up. Again, if you want to go through the specific genetics work that they've done here, like their actual methodology, it's here at the end. And they talk a lot more about the specific loci of some of the genes that they're looking at, which again is all kind of over my head. The specific evolutionary pressures relating to the loss of the tail in hominoids are not clear, although they are probably involved in enhanced locomotion and the transition to a non-arboreal lifestyle. Again, I disagree for reasons that I took a long time to explain earlier in this video. We suggest, however, that the selective advantage must have been strong because the loss of the tail may have included an evolutionary trade-off of neural tube defects, as demonstrated by the presence of neural tube closure defects in mice expressing the TBXT exon 6 transcript. Notably, mutations leading to neural tube defects and/or sacral agenesis have been detected in the coding and non-coding regions of the TBXT gene. We therefore speculate that the evolutionary trade-off involving the loss of the tail, made approximately 25 million years ago, may continue to influence human health today. Opening up an entire new niche for an organism to exploit resources within, I think, is a pretty strong selective pressure, so it's not surprising that there is a potential downside to the taillessness that we see in hominoids. And I'm curious to see if there are similar consequences in other primates that have lost their tails. Hopefully work is being done on that as we speak. But yeah, I think this paper was super duper cool. It's a great example of how predictions work and come to fruition in evolutionary biology, given the ALUs that they were looking at have been hypothesized to be potentially responsible for quite some time. And now they've got the experimental support to kind of prove it. I mean, you don't prove anything in science, but you kind of get what I'm saying here. I think we have a pretty concrete how and why as far as the loss of the tail in hominoids, which means we're this much closer to understanding our entire evolutionary lineage in full. We'll get there someday, I know it. The results of this paper also make it seem like it was a pretty quick transition. It just goes to show that big phenotypic change can be managed by a relatively small genotypic tweak. Anyways, my gentle and of course very modern apes, I thought this was a cool paper. I hope you thought it was cool too. I know we really didn't go over the paper. The paper was mostly like a cherry on top of me waxing poetic as to my favorite hypothesis for why hominoids don't have tails, but I hope you had fun nonetheless. If you like what I do, please consider supporting me in the free way. It is of no cost to you to like, comment, and subscribe. I don't think that's of any cost to you unless you just really hate seeing my mug on your home screen. And if you want to support me in greater ways, you can join my Patreon. Folks who are a part of the Patreon sometimes get early access to videos and they get their name at the end of every single one. Unless I've missed one, I don't think I've ever missed any, so let me know. Sometimes I lag behind too, like the video gets completed and then it's waiting to be released and then more people join, so you'll have to excuse me on that. Just little things, you know, I like to cover my bases. And in the meantime, I hope to see you very soon next time.